Hi, welcome to BugSigDB.org, a manually curated database of published microbial signatures. In this video tutorial, we demonstrate how to use the database, how to search for studies by different criteria, and how to export information from BugSigDB for incorporation in a downstream analysis. In two separate video tutorials, we also explain how to add a study through the curation interface of BugSigDB.org and how to work with BugSigDB data in R using the BugSigDB R bioconductor package. We start with an overview of the contents linked on the main page of the database, and after that, introduce different entry points for the exploration of individual studies. The main page of BugSigDB.org provides basic information about the project, including how to request an account in order to contribute to the project, how to provide feedback, and also how to report issues with functionality on BugSigDB. For more information, we can visit the About the Project page, which contains more information on what BugSigDB is, on how BugSigDB can be cited, and how to contribute to the project, including detailed information for curators. This includes information on how to request an account and where new curators can get started to contribute. Besides general information about the project, the main page also provides different entry points to the curated information in BugSigDB. This includes retrieving signatures by a certain taxon of interest, browsing curated studies by name, and basic and advanced searching functionality, which we will introduce later in this tutorial. In addition, the page lists comprehensive summary statistics of the database contents, which at the time of recording this tutorial contained more than 2,500 microbial signatures from more than 700 microbiome studies. We proceed by exploring the anatomy of BugSigDB study pages, using here for demonstration a study on autism, which was published in 2018. BugSigDB study pages can be roughly divided into three main sections. A first section that provides basic information about the study, an experiment section that provides detailed information about how microbiome measurements were obtained and analyzed, and a third section that contains signatures of differential abundance, each listing one or more microbial clades. We now take a closer look at what information each of the three sections provides. The study section categorizes studies by study design, and each study is associated with a study identifier, such as the PubMed ID or a DOI, depending on whether a study is indexed in PubMed. The experiment section records key characteristics about the profiled subjects, about lab analysis procedures, about statistical analysis, and alpha diversity measures. For the subjects, this includes the geographic location, the host species from which microbiome samples have been obtained, the body site, the study condition of interest, as well as antibiotics exclusion and sample size in both the study group, group 1, and the control group, group 0. To define the two sample groups that are contrasted for differential abundance, BugSigDB also records the diagnostic criteria applied to define the specific condition represented in the study group, in the group 1. Of particular importance are the body site and condition fields, which are standardized based on established ontologies. The Uberon anatomy ontology for body site and the experimental factor ontology for condition. This makes it easy to identify other studies investigating a certain condition, here autism, by clicking on the corresponding condition term, which leads us to a dedicated condition page for autism that also links all other studies in BugSigDB that are investigating autism. On the other hand, this also allows us to leverage ontological relationships to perform high-level queries against BugSigDB. For example, if we follow the link to the experimental factor ontology for autism, we find that autism is classified as a nervous system disease and as a psychiatric disorder. This classification allows us to not only obtain these signatures when searching explicitly for autism, but also obtaining these and other signatures by searching for either nervous system diseases or psychiatric disorders. The signature section typically contains for each experiment 2 signatures, a signature of increased abundance in the study condition and a signature of decreased abundance in the study condition when compared to healthy controls. Microbial clades annotated to a signature are standardized based on the NCBI taxonomy, facilitating lookup of a taxon in the reference database, here displaying the phylum Bacteroidota. On the other hand, this standardization also provides for swiftly identifying other signatures containing that taxon in BugSigDB. Here we are seeing all studies and associated signatures that either contain the phylum Bacteroidota directly or certain descendants of the phylum Bacteroidota, such as the genus Bacteroides or Elistipes. We also note that the contents linked on a study page are associated with quality flags, which indicate the curation status of the content. This quality flag could be either stating incomplete, which would indicate that the content is currently under active curation. This quality flag could also state complete, which would indicate that the curator has completed the curation of this content, or, as we see it here, the quality flag could also state reviewed, which indicates that another curator has reviewed and approved the curated contents. As we explain in the curation tutorial, it is also possible to edit existing content or flag content for review if certain quality issues have been identified. This will, however, require to sign up for an account. There are 3 main routes to search for contents on BugSigDB. The general search bar to search for specific keywords of interest, the taxon lookup that allows to identify all signatures containing a taxon of interest, and the advanced search that provides for the highest level of customization of the search query. A typical use case for the general search bar is to quickly identify studies investigating a specific body site or condition of interest. For example, I could here search for all studies investigating breast cancer, which would lead me to a dedicated condition page for breast cancer, as we have seen before for autism, that also links all studies investigating breast cancer in BugSigDB. The taxon lookup bar is useful for quickly identifying all signatures that contain a taxon of interest. For example, I could here search for the genus Escherichia, which would lead me to a page, as we have seen before, for the phylum Bacteroidota. Note that the search result typically consists of 2 parts. On the one hand, all signatures that contain the genus itself, called here containing this taxon directly, and on the other hand, all signatures that contain descendants of that genus in the taxonomic hierarchy. Here, for example, the species Escherichia coli. The advanced search allows to parameterize the search query further by selecting values for one or more of the main fields of interest. For example, this allows to quickly identify all studies from the United States that are investigating human subjects that obtained stool samples and that were investigating colorectal cancer. We could also specify certain taxa of interest which we would like to be present in the signatures satisfying these search criteria, which we, however, omit here. The result page then contains all studies that were investigating colorectal cancer for the subjects satisfying these search criteria, and it would be possible to download these studies as plain text files given the study information, the experiment information, and the corresponding signatures. In addition to exploring microbial signatures directly on BugSigDB.org, it is also possible to download BugSigDB data in bulk for incorporation into other applications and downstream analysis. There are different options for obtaining the full BugSigDB data dump. The first option is to download the full unfiltered data dump directly from BugSigDB, which will also include content that is currently under active curation. The second option is to obtain a weekly snapshot from GitHub, which will include the data dump that has been automatically filtered to only contain complete content. The preferred option for most users, however, will be to obtain one of the official releases of BugSigDB data from Zenodo. These data releases have been automatically checked and manually reviewed, providing for a high-quality data dump. For all users that wish to analyze BugSigDB data in the R programming language, it will be most convenient to obtain BugSigDB data directly from within R, using the BugSigDB R bioconductor package, which we will demonstrate in a separate tutorial. The recommended approach for obtaining the latest release version of BugSigDB is to download the data from Zenodo. This is especially relevant for all users that wish to incorporate BugSigDB data into analysis with third-party software, such as gene set enrichment analysis software. From a closer inspection of the files contained in a BugSigDB data release, we see that there are two parts. One CSV file that contains the full dump of the database, including all curated fields for studies, experiments, and signatures. And the second part that consists of various GMT files that contain only the microbial signatures in different formats with regard to the taxonomic level and the taxonomic ID type. The GMT file format is a standard format that is compatible with all major gene set enrichment analysis software, and a user can choose here from obtaining signatures on the genus level, on the species level, or signatures containing a mixture of taxonomic levels as typically found in the literature. On the other hand, a user can choose to obtain signatures using NCBI taxonomy identifiers for each taxon, the full taxonomic classification for each taxon in metaflan format, or the taxonomic name of each taxon only. For signatures harmonized to the genus level or to the species level, it is possible to either obtain signatures that are restricted to taxa on the genus or species level only, or signatures that will also contain taxa that are found at a more specific taxonomic level by cutting the taxonomic tree at either the genus or species level. As we mentioned before, it is also possible to obtain weekly snapshots of the database from GitHub, from the BugSigDB exports GitHub repository. As the official releases on Zenodo, the weekly snapshots on GitHub contain the full dump as a CSV file and the microbial signatures on different taxonomic levels in GMT file format. In contrast to the release on Zenodo, however, the weekly snapshots have not been manually reviewed. On the other hand, users can also download the full unfiltered data dump from BugSigDB.org directly, which consists of a CSV file for study information, experiment information, and the annotated signatures. Users should keep in mind, however, that this unfiltered data dump contains incomplete content that is under active curation and that should be filtered out prior to any analysis.